Another important topic which we should learn is the tricarboxylic acid cycle, TCA cycle, or citric acid cycle, which are differently named this way. But before going into the TCA cycle, we should learn what happens to the pyruvic acid that is formed due to the process of glycolysis. Now, as we all know that glycolysis is the process whereby glucose is oxidized to pyruvic acid in the aerobic condition, and in the anaerobic state lactate is formed. Now, the pyruvic acid in the aerobic condition has to be converted to the acetyl coenzyme A. Now, pyruvic acid is formed in the cytoplasm, and this has to enter the mitochondria by special transport systems into the mitochondria, and inside the mitochondria it is acted upon by pyruvate dehydrogenase complex. It's a complex system of enzymes with 3 enzymes. Now, the 3 enzymes are pyruvate dehydrogenase itself, and then trans-lipoyl acetylase and trans-lipoyl dehydrogenase. So there are 3 enzymes, and it also involves 5 coenzymes. The coenzymes are coenzyme A, you have lipoic acid, you have then thymine pyrophosphate, you also should have the FAD and the NAD. So there are 5 coenzymes used, and there are 3 enzymes. Now, with the help of these 3 enzymes and the 5 coenzymes, the pyruvic acid is converted to acetyl coenzyme A within the mitochondria. Now, this acetyl CoA, the fate of this acetyl CoA is to join with oxaloacetic acid and form the citric acid with the help of the enzyme citrate synthase. Now, this is what is happening in the aerobic state. But in the anaerobic state, the pyruvate will be reduced with the help of NADH to lactic acid. Now, lactic acid can be converted to glucose later also. Now, and so it serves as a gluconeogenic substrate. Now, the excuse me, the there are some special aspects of glycolysis in RBC where the glycolysis always ends in lactate. But now, talking more about the TCA cycle, the TCA cycle starts with the formation of citric acid with the help of oxaloacetic acid and acetyl-CoA. Now, the citric acid is converted to citric acid, and then it is converted to alpha ketoglutaric acid. And this alpha ketoglutaric acid, it is a keto acid like pyruvic acid, and with the help of alpha ketoglutaric acid dehydrogenase, which is again a multi-enzyme system, and with the help of this multi-enzyme system having 3 enzymes and 5 coenzymes, analogous with the pyruvate dehydrogenase complex, the alpha ketoglutarate dehydrogenase is converted to succinyl coenzyme A. Now, succinyl coenzyme A has got a high energy thioester bond, and this thioester bond can be the energy of the thioester bond can be incorporated into GDP plus PI, so that GDP can be synthesized in the next reaction, and hence a substrate level phosphorylation takes place at this point of time. Now, the enzyme is succinate thiokinase. So, with this, the succinyl-CoA is now converted to succinate, and then succinate becomes fumarate with the help of succinate dehydrogenase, and fumarate is converted to malate, and then malate is further converted to oxaloacetic acid with the help of malate dehydrogenase, which requires NAD. Now, succinate dehydrogenase requires FAD, which becomes FADH2. Now, it can be seen that during the process of TCA cycle, there are 3 NADH molecules and 1 FADH2 produced, and there is also one substrate level phosphorylation, that is when the succinyl-CoA is converted to succinate. So, the 3 NADH molecules can further go to the electron transport chain, and from each NADH molecule we can get 2.5 ATP molecules, and from the 1 FADH2 molecule 1.5 ATP molecules can be generated. So 2.5 into 3, that is 7.5, and then 7.5 and 1 FADH2 molecule, that is 1.5, 7.5 plus 1.5, that is 9, and then 1 substrate level GTP. GTP is formed, not ATP, but GTP. So totally we can get 10, that is 9 plus 1, 10 ATP or high energy phosphate bonds in one turn of the cycle. Now there will be 2 acetyl-CoA molecules produced from one glucose unit, and so we get 10 into 2, that is 20 ATP molecules in the TCA cycle, along with the electron transport chain. So quite a lot of ATPs can be synthesized in this manner. So this is now you can also see that TCA cycle is the final common pathway for all the metabolisms to converge. That is, glucose metabolism enters the TCA cycle as pyruvate and then acetyl-CoA. The lipid metabolism done with the help of beta oxidation produces acetyl-CoA and then enters as acetyl-CoA from the fatty acids. I mean, if there is a fatty acid of 16 carbons, then it can produce 8 acetyl-CoA molecules, and then it can go into the TCA cycle. Now, similarly, the proteins also get hydrolyzed to amino acids, and various amino acids, after removing the amino group, will be left with the carbon skeletons, and they enter the TCA cycle at various points. For example, aspartic acid, by removal of amino group, it will enter at the level of oxaloacetic acid, whereas glutamic acid, by the removal of amino group, enters the alpha ketoglutarate dehydrogenase step in the form of alpha Kg actually. So and the pyruvate can be obtained from alanine. So like this, and succinyl-CoA can be obtained from various branched chain amino acids. That shows that various amino acids, after transamination and deamination, are entering the TCA cycle as the final common pathway. Now, final common pathway, of course, has got another pathway, that is the electron transport chain also. So we will be taking up the electron transport chain at a later time. So that is about the TCA cycle and its importance. And another importance is that it is an amphibolic pathway. That means both the the anabolism and the catabolism is going on. Catabolism in the sense the amino acids are all transaminated and deaminated into the intermediates of the TCA cycle, whereas the anabolism will take place when the pyruvic acid is transaminated to alanine with the help of ALT enzyme or GPT enzyme, and alpha ketoglutarate will be transaminated with maybe with the help of the ALT itself to form the glutamic acid, and oxaloacetic acid can be transaminated to form aspartic acid in the presence of AST, that is aspartate transaminase. Now in this manner it is catabolic and anabolic also, because amino acids can be synthesized, that is the non-essential amino acids can be synthesized. We cannot synthesize the essential amino acids, but only non-essential amino acids can be formed in this manner. Now TCA cycle also is a helping in the gluconeogenesis, because the gluconeogenesis can be done from glucogenic amino acids like glutamic acid, aspartic acid and pyruvic acid, because by a backflow from alpha ketoglutarate dehydrogenase, alpha ketoglutarate to oxaloacetic acid, and then oxaloacetic acid can be converted to phosphoenol pyruvate, and phosphoenol pyruvate can be converted to fructose 1,6 bisphosphate, and then to fructose 6-phosphate, then to glucose 6-phosphate, and further glucose 6-phosphate can be converted to glucose. So that's why the TCA cycle is known as the amphibolic pathway. It is the final common pathway where all the fuels, that is the carbohydrate, proteins and lipids, they are converted to some of the intermediates of the TCA cycle. So the deficiency of the enzymes of the TCA cycle is not compatible with the life, because if the TCA cycle enzymes, or for that matter the electron transport chain enzymes or the steps are defective, then all the three metabolisms will be affected, that is the carbohydrate, protein and lipids will be affected, and the energy production becomes difficult, and so the life is not possible. So this is about the TCA cycle enzyme. Now another important metabolic pathway in as far as carbohydrates are concerned is the hexose monophosphate shunt pathway. Now this HMP shunt pathway does not produce any energy. It simply produces the reducing equivalents in the form of NADPH, and also it produces ribose 5-phosphate. Now glucose 6-phosphate is required for this process. The glucose 6-phosphate can be converted to 6-phosphogluconolactone in presence of NADP and glucose 6-phosphate dehydrogenase enzyme. Now this is forming, that is the 6-phosphogluconolactone. In the next step, 6-phosphogluconolactonase enzyme adds water, and then it is further converted to the next compound, and then there is another step where there is a second dehydrogenation, and here also the NADP is the acceptor of the hydrogen, and hence you get NADPH. Now NADPH, two molecules of NADPH are produced in this pathway, and later stages there is a production of ribose 5-phosphate, and ribose 5-phosphate is important in the formation of purine and pyrimidine bases, and also the formation of nucleotides mainly the formation of nucleotides, purine and pyrimidine nucleotides, which are important for the synthesis of ribonucleic acids and deoxyribonucleic acids also. This NADPH, on the contrary, has very many important functions. NADPH is required for the synthesis of fatty acids in the body, and NADPH is also required for the synthesis of cholesterol in the body, and the conversion of cholesterol to bile acids, bile salts, or conversion of cholesterol to the steroid hormones also require NADPH. So NADPH is a very important reducing equivalent or coenzyme that is of importance. Now, in the another importance is that this NADPH maintains the integrity of the RBC membrane. Now, due to the production of H2O2, hydrogen peroxide, the hydrogen peroxide should be removed as it is a toxic substance, and this is done by the glutathione, that is the reduced glutathione. So glutathione converts the hydrogen peroxide into water, and during this process the glutathione reduced becomes oxidized, and the enzyme for this reaction, that is the glutathione peroxidase. Now glutathione oxidized has to become glutathione reduced, and the enzyme is glutathione reductase, and this requires NADPH. Now NADPH is provided by the HMP Shunt Pathway, that is the G6PD enzyme. So the glutathione oxidized can become reduced. Now the G6PD enzyme may be deficient in some individuals, so care should be taken when administering certain drugs like anti-malarial drugs or sulfur drugs or aspirin to these patients, because as these drugs are oxidants and might produce the free radicals, and for the neutralization of the free radicals, the glutathione is required. Reduced glutathione is required. Now, reduced glutathione level is maintained with the help of NADPH. So, if there is a deficiency of G6PD enzyme, the NADPH production is decreased, and hence necessary NADPH is not available for the reduction of oxidized glutathione to reduced glutathione. Hence, the H2O2, which might increase in the RBC, may convert the hemoglobin into methemoglobin and may lead to hemolysis, and then this may lead to hemolytic anemia and hemolytic jaundice. So there are two states. G6PD may be half present, or it may be fully absent. So those with G6PD enzyme to the half the level is known as the heterozygous state, and the complete absence will lead to the homozygous state. So heterozygous people can live a normal life, provided that they are, while administering drugs, proper care is taken to this effect. So, another function of NADPH is to reduce the methemoglobin to the reduced hemoglobin. So, for this also NADPH is very much required. And for the detoxification of drugs, also NADPH is important. So, NADPH has several important functions, which is produced mainly by the HMP shunt, and also isocitrate dehydrogenase, which is present in the cytoplasm, also produces NADPH, whereas the ICDH, that is isocitrate dehydrogenase, present in the mitochondria, uses NAD as the coenzyme. So NADPH is produced from cytoplasmic ICDH also, and another enzyme, malic enzyme, also produces NADPH to some extent. But the main contributor of NADPH is the hexose monophosphate shunt pathway. And as it is required, as NADPH is required for steroid hormone synthesis and also fatty acid synthesis and also cholesterol, those organs which synthesize steroid hormones, cholesterol, fatty acids, all will have a functioning NADPH production or the HMP shunt pathway. Tissues like lactating mammary glands and gonads and adipose tissue and liver have actively functioning HMP shunt pathway, and also the RBCs are important to have the HMP shunt pathway so as to prevent the hemolysis. So this is the significance of HMP shunt pathway.